Right. Hi, everybody. I'm Matthew Venner. I am, as Daniel said, a recent graduate of the University of Otago, and I did my PhD on the fossilised wood of New Zealand. Um, Basically, what it entailed was taking material from about 20 different field sites scattered throughout the South Island of New Zealand, the Chatham Islands, as well as the Auckland Islands. And so I'm going to give a quick overview of basically the results from that study. So a quick outline for this talk. I'm going to give a bit of background on how to actually identify wood, as well as previous work in the field. I'm going to then talk about the fossilised wood of New Zealand through time, the fossil forest components, and then I'm going to end with a little bit of a discussion on the paleoclimate and physiognomy and how you can actually try and tease out paleoclimate signals from wood anatomy itself. So firstly, why would we want to study fossilised wood? Well, as most people can tell you, it is incredibly abundant. It can be identified to mostly usually family genus level. Once in a while, you might be able to identify it down to species level as well. It is currently in New Zealand underutilised as a source of paleoclimate and paleoecological data. We don't really have anybody who previously have used this this material as a form of paleoclimate studies. And this material can be used in conjunction with data from other plant fossil material like leaves or seeds or pollen or what have you. So a really quick foray into the identification of wood for those of you who don't know much about it. So very basic botanical nomenclature. Conifer, Gymnosperm, those are the cone-bearing plants like Totara or Kauri. Angiosperm are our flower-bearing plants. They both have very different types of wood anatomy, as we will soon see. Just an overview about fossilised wood in New Zealand. In New Zealand, we have fossil wood from as old as the Permian. Unfortunately, I was not able to actually get my hands on any material that was this old, and so my my studies were limited to just as old as the mid-Jurassic. There's a variety of preservation styles, and each of those different preservation styles, whether they be replaced by silica or replaced by calcite, or not really replaced by any sort of material at all, require a slightly different method to actually be able to section and then tease out that important cellular information. So this is just a kind of a really brief overview of some of the fossil wood material that you can find in New Zealand. You can see that Morgan's Hollow material. You have material ranging all the way from this really dark material that kind of almost looks like just to this really light colored material that almost, if you look at it, looks like some sort of quartzite or other material like that. You have the Silcrete material from Lansip Hill. You have this really dark agatized material from Mount Somers at Canterbury. And you have material that has basically not been replaced by any sort of mineral at all, like from Idaburn and Otago. Now on to the actual wood identification part. To identify wood, this goes for both fossil and modern. You need to be able to view the wood anatomy from three different orientations. The transverse, the tangential longitudinal, and the radial longitudinal. As you can see, the transverse section is that really typical kind of ring view. When we're looking through the tree, we can see the nice growth rings. The tangential longitudinal is basically, if you imagine a circle taking a radius from the center of that circle out, that would be your your radial longitudinal, and your tangential longitudinal is at right angles to that tangential longitudinal. So just talking about conifers versus angiosperms, I said before that they had quite a different look between them. And if you look at these two transverse sections, you can see that that is quite so. Conifers basically only have about 3, sometimes 4 main cell types. Those are the tracheids, the rays, the axial parenchyma, and if you're not in New Zealand, you have a few other weirder types of cells. Whereas in the angiosperms, you have a much more diverse range of cellular types, including vessels, once again, tracheids, axial parenchyma, rays, but you also have fibers and a number of others on top of that. So the main features of a conifer, the conifers in the transverse section, might be able to see axial parenchyma, which you can see are these kind of dark, square-shaped cells. The tracheids, which make up most of the rest of that, are these hollow square cells, and then the rays are these kind of vertically aligned, in this case, black lines. They can have growth rings, they can not have growth rings, and they also have radial parenchyma, which in New Zealand tend to be unicellular, meaning they are only really one wide and tall. The really important thing for identifying conifers is tracheid pitting. So tracheid pitting has a number of different ways and patterns that can be going, including, in this case, you can see this is a pit here and this is a pit here. These ones are directly opposite each other. You can see that this here is a pit and this here is a pit. Those are alternating in between each other rather than being opposite. You can see, and with these two, those two tracheid pits are separate from each other, whereas in this picture they are contiguous. They are joined together at the hip. And you can get uniseriate pits, so pits in columns of one, biseriate, so pits in columns of two, or triseriate even are pits in columns of three. The other important feature for identifying conifers is crossfield pitting. You can see crossfield pitting is essentially the crossfield, where if you have a tracheid, which is in this case vertical, and a ray, which would be that horizontal light horizontal line, those are the pits that go directly in between a ray and a tracheid, and those have a number of different features and different patterns as well. So when I was identifying the conifer material, I always referred back to a paper by Philippe and Bamford in 2008, which essentially listed all of the Mesozoic conifer genera that have so far been identified today. In contrast, when I was identifying the Cenozoic material, I would relate it back to mostly the modern conifers actually growing in New Zealand today. So just a few examples of the various types that we can find. Here we have Araucarioxylon and Araucarioxylon. The main features that we have here are these contiguous alternating tracheid pits, as well as these contiguous alternating crossfield pits, as well as uniseriate rays. In the polycarps, we have separate uniseriate to occasionally biseriate rays. We have axial parenchyma as well as rays. We don't really have axial parenchyma in the Araucarioxylon. And the crossfield pitting, there's usually only one, sometimes maybe 4 pits per crossfield in comparison to the Araucarioxylon, where we can have up to 16 pits per crossfield, which is why these ones look quite so crowded. Right, on to the angiosperms. So angiosperms, in comparison to conifers, you have those 3 cell types, and they have maybe 3 variations amongst them. In terms of angiosperms, we are looking at more like 200 variations of the various cell types, which means that the actual identification of an angiosperm is much more involved and can be much more complex and time-consuming. So vessels, just as a start, one cell type, when we're looking on the transverse face, can be solitary. They can be grouped in radial multiples. They can be grouped in tangential multiples. They can be grouped in festoons. They can be grouped in random clusters. So that's 5 or 6 just variations on how vessels are grouped. That's before we even start looking at their size, their shape, and what have you. The next thing that we look at when we're looking at angiosperms is perforation plates. Now, perforation plate is essentially when you have the end of one vessel strand that connects to the next vessel strand above or below. And that can either be simple, in which you just have a really simple circular hole, as you can see in this picture here, or it can be scalariform, in which you have this kind of bar-shaped cell in between them, in which case you would see almost a ladder structure in between in that circle. Angiosperms also have a wide range of rays. So rays can be uniseriate all the way up to 10 plus seriate, so 1 cell wide to 10 or more cells wide. That's what you can see here. This is a uniseriate ray. This is one of those large multiseriate rays. They can also be aggregates, in which you have a bunch of smaller rays which have been aggregated together. And in hand specimen, this ray looks like a single ray, but when you look at it underneath the microscope, it actually looks, you actually realize it's actually more than one ray. Angiospermous identification was basically done by looking at the IAWA database, the Inside Wood database, which has, the last time I checked, something like 30,000 or ballpark figure species which have had the various cell types all mapped out and put online, which you can then quite easily go and search if you have the various features that you want. I then, after doing that kind of brief study, go and look further into those results of that search to see if any of them are actually anything sort of likely. So when I'm talking about the Inside Wood database, you are able to search for a series of characters, and those characters, as one of, the 200 or more species possible. I usually do multiple searches using different features, just so that I know that, okay, what I'm actually pulling up is making sense. 221 features listed, but not all of them are applicable to fossil. You've got things like heartwood color, you've got things like scent, that sort of stuff, which doesn't really work with a fossil wood that's been dead for 180 million years. So as an example, this is an angiospermous sample of wood. You can tell that just in hand specimen because you can actually see the vessels if you look close enough. I've made slides from 3 different orientations, and this wood has diffuse porous vessels. So the vessels are not grouped in any way. They are diffuse, so they aren't related to a tree ring in any way. They have a kind of broadly diagonal pattern. You can see kind of going down in this direction if you can follow my mouse. They're solitary, and there's also these weird kind of cell interruptions called tyloses, which you can kind of see makes up this weird black matter which is in the center of these vessels. In this sample, the rays are uniseriate. All the ray cells are procumbent. It means they're lying on the side. You can see here they kind of look like they're oblongs but lying on their side, and you can see that you have fibers with quite distinct pits. So a search on the Inside Wood database for a diffuse porous wood with vessels in a diagonal or radial pattern, which are also solitary, tyloses, common uniseriate rays procumbent, and fibers with distinctly bordered pits on both the radial and tangential walls, basically looks like this on the database. And when you click the 0 mismatches allowed and then hit the search modern hardwood button, you come up with 8 samples, 8 possible species which this sample could be related to. And if we look here, we've got everything from Fabaceae to Fagaceae to Myrtaceae to Scrophulariaceae. Some of these are obviously not in New Zealand. In fact, most of them are not in New Zealand. Oddly enough, we have this eucalypt. There are some people who don't really like that result, but oh well, it's fine. Anyway, so that's how you identify, or how I have identified an angiospermous wood. And now I'm going to go on to really quickly what previous work has been done in this area. So Mesozoic previous work goes all the way back to the late 1800s and early 1900s with people like Creagh and Edwards, who worked on material from the South Island from the Catlins, as well as other people who've worked on material from Port Waikato, Vanessa Thorne, who some of you may know. They identified a number of coniferous genera, including Araucarioxylon and Podocarpaceae. In the Cretaceous, we have once again some reaching back into the late 1800s, von Ettinghausen working on Araucarioxylon from the Melbourne Hills, and Marie Stopes working on Araucarioxylon and Planoxylon from kind of the upper coast of the South Island. Sorry. And we also have a more recent work by Paul and Felipe on the Taxodioxylon of Pitt Island, part of the Chatham Islands. Moving into the Cenozoic, we have Araucarioxylon from the Paleocene and Wongaloa. There's not really too many other sites that have fossilized wood that is well enough preserved from the Paleocene to actually do a study on. From the Eocene, we have Exerba from Pico Pico and Podocarpaceae and Araucarioxylon from two different sites, Pico Pico and the Waihau River. In the Oligocene, we've only got this one kind of site in Cozy Dell that has yielded some polycarp wood. The Miocene is really where our amount of knowledge of the fossilized wood flora of New Zealand really kind of starts to get a lot. You have in the North Island, Sutherland did a study on Kaipara Harbour and found a wide range of Angiosperms genera, including Avicennia, Nidophagus, Myrtaceae, Vitex, etc., as well as conifers, while Moore and Wallace looked at the Coromandel material and found Casuarinoxylon, Nidophagus, Weinmannia, amongst others. And from the South Island, we have conifers from the Gore lignite measures. We have Casuarinoxylon from Lancip Hill. We have Nidophagaceae from Roxburgh. We have more conifers from also Roxburgh and Idaburn, Casuarinaceae from St. Bethans, and Myrtaceae from Lake Aviemore. You'll notice that a number of these have my surname on them. So this is what this map looks like before I started my PhD study. This is what the map of studied sites from New Zealand looks like before I started my PhD. And that's as we move into the current study, hopefully we'll see a slightly more full map after that. So my current study, I looked at 22 field sites from the South Island, as well as Waihiri Bay and on Pitt Island and Fairchild's Garden on the Auckland Islands, apologies. And I looked at more than 100 new samples. Not all of them, however, were identified. Occasionally you pick up a sample which just isn't well enough preserved to actually be able to say anything relevant about it. And this study includes both new coniferous and new angiospermous material. So there is that map of material where material has been studied previously, and here it is now after I finished my PhD. This is a much more comprehensive overview, if I do so myself. So from the Jurassic, I identified material from 3 different sites: Cleet Hills, Productus Creek, and the Catlins. Now, the Productus Creek material actually might be Permian, but we just wanted to go with the safe option of calling it Jurassic. At the Cleet Hills, I identified this weird coniferous or Mesozoic conifer called Planoxylon. At Productus Creek, I identified Agithoxylon, which I also identified from the Catlins, along with this type C Mesozoic conifer, which is basically just a conifer that has polycarpoid features, but cannot be related to any specific Mesozoic coniferous genera. So this Planoxylon is quite strange in that it has features which are both reminiscent of both Araucarioxylon as well as a Podocarpaceae. You can see it's got these contiguous and opposite tracheid pits, but it also has separate pits. In this case, they are actually alternating clustered pits, pits which are also opposite and separate. And then it has, so those are the features that it has, that it shares with Araucarioxylon and Podocarpaceae. It also has its own really special feature, which is a long German word, which essentially means radial pitting. You can kind of see that radial pitting in here with these bulbous kind of outgrowths in the rays, as well as in the tangential face, where you have these pits right in the center of the cell. Planoxylon has an interesting history. It was first identified from New Zealand by Marie Stopes, but this is the first time, however, that it has actually been identified from the Jurassic rather than just the Cretaceous. Speaking of the Cretaceous, we have a number of different sites, which is why we have Cretaceous 1. From the Conway River, we have Planoxylon and that type C Gymnosperm. From Mount Somers, we have Angiosperms. From Awaro, we have both Angiosperms and Planoxylon. And from Port Robinson, we have this type C Gymnosperm. Now, all of these sites probably yield more material, which will give additional species or genera. From the Quail Flat, we have the type C Gymnosperm. We have from the Royal Mine Casuarinoxylon, the first time this specific genus has been identified from the mainland of New Zealand. And from Whakapohai, we have Arececeae, and from Waihiri Bay, a large study yielded Arececeae, Taxodioxylon, Protophylloclodoxylon, and Cupressinoxylon. Now, those two haven't actually been identified from Zealandia at all in the past. Protophylloclodoxylon and Cupressinoxylon. So this is just kind of if we're just looking at the material from Pitt Island, and this is the range of different cell types that we get just in conifers from Pitt Island. So we have Cupressinoxylon, Protophylloclodoxylon, Taxodioxylon. They all have a really wide range of different cell types. Protophylloclodoxylon, Taxodioxylon both have quite a lot of axial parenchyma, or as well as Cupressinoxylon has more of this. It has these axial parenchyma and kind of tangential bands. The range of tracheid pitting is quite diverse, as is the range of crossfield pitting through all of these. We kind of unfortunately missed the Paleocene. I didn't make sections of any new samples for any new species, so we go straight into the Eocene. From the Eocene, I managed to sample from two sites, from Boulder Hill, which is right up into Dunedin, the Dunedin town zoning. I identified a fossil legume, which is quite probably a new genera in itself. A new genus in itself. And from Hampton Beach, I identified a monocot wood, which is possibly a palm, but unfortunately, due to its preservation, it doesn't have all of the features required to definitively say that yes, this is a palm, and then say which genus that palm, what species that palm should be. So the legume sample has a very wide range of features. You have these vessels, which may be solitary, but they also may be in these kind of irregular clusters. You have axial parenchyma, which is in these long kind of tangential bands, as well as in these kind of wing-shaped loops around the vessels. You have incredibly minute intervessel pitting. So this is the pitting in between two adjacent vessels. You have rays which are both procumbent, so lying on its side, but they also have a few upright cells. You have rays which are almost always three series, three in a row or two in a column. You have these simple perforation plates, and you have these really tiny vessel ray pits. Moving on to the palm, you can see that this palm has the really typical kind of, I've seen this described as a mushroom cap or a happy face shape with a vascular bundle there, as well as these scalariform type perforation plates in between that vascular bundle. Moving on into the Oligocene, we have Gymnosperms and Angiosperms material. From Cozy Dell, I found an undetermined angiosperm. Now, unfortunately, this material didn't actually have enough features to be able to refer back to what angiosperm it actually might be. But we can definitively tell it is definitely an angiosperm, and that's quite nice just to know that we had angiospermous wood in New Zealand at this time. And from Pomahaka, I identified Agathoxylon type wood. So here is that undetermined angiosperm. And now you can see it's got these scalariform perforation plates. So it's undeniably an angiosperm. However, where those vessels actually are in the transverse space, I have genuinely got no idea. I cannot see anything that in this transverse space actually looks like what a vessel should look like. So this is being crushed, modified, and just completely destroyed, almost beyond recognition, with the exception of occasionally one of these really nicely preserved vessels in the radial phase, which actually has these scalariform perforations. From the Miocene, I identified a number of Gymnosperms, including Podocarpaceae and Araucarioxylon, and for the first time, I was able to possibly say, determined that we had both Agathis and Araucaria. Previously, Araucarioxylon type wood really doesn't have many macro features which enable it to be easily determined between the three different species within the Araucarioxylon. However, a paper came out recently which showed that a few different features may actually be used to be able to separate them. And so I used those features and showed that there was a possibility of having both Agathis and Araucaria in the New Vale coal mine area. The angiosperm material was considerably more. I kind of focused on angiosperm material for my PhD, unlike my master's, which found more coniferous material in the Miocene. So we have from the Auckland Islands, Araliaceae, from White Rock Quarry, Lake Aviemore, and the Ashley River Eucalyptoxylon, so a fossil eucalypt. If you look at the distribution of those three sites, you have White Rock Quarry, Lake Aviemore, and the Ashley River, and it's actually quite a wide range of that. So we possibly had a reasonably large forest of eucalypts in the Miocene, or early Pliocene. From Burnside Quarry, I identified Casuarinoxylon. This kind of is in conjunction with the material that I identified for my master's from Lansip Hill, which is also Casuarinoxylon, but possibly a different species. And from Karatani, I identified a possible Malacytis, but its actual identification is a little bit in doubt. So here is that fossil Araliaceae from the Auckland Islands. When I picked up the sample, when Marcus first showed it to me as material that came from the basement here, I didn't really know what to do with it, and I thought that I would probably pass on that material. However, through a series of fortuitous events, we were able to find the person who found it, and we were able to tell from that person who collected this material back in the late 1970s that he had collected it from the Auckland Islands. And so, I thought, I have to study it now. And so I was able to get some actually really nice sections, despite how poor quality the wood initially looked. And you can see it has kind of separate solitary vessels, but you also have random clusters of vessels, as well as these kind of tangential bands. It has kind of between 5 and 10 seriate rays, which are incredibly tall. These rays can be millimeter scale tall, which surprised me quite a lot. Really nicely preserved scalariform perforation plates, quite nicely preserved vessel ray pits, and other intervessel pits of various types. So this sample actually really surprised me as to how well preserved it was, because if you look at the sample, it looks like a kind of like a piece of poorly preserved lignite that's been sitting out on the surface of the ground for some time and has been really badly eroded. So I took some Aralia, some actual modern lancewood material from the Auckland Islands. I was lucky enough to have to gain a permit to send someone down there to bring some back for me, as well as have some sent down from Landcare. And you can actually see there's quite a few differences in between the modern Araliaceae, the modern lancewood material and the fossil Aralia material, the fossil Araliaceoxylon. Firstly, instead of being kind of all dispersed all the way throughout the growth ring, the modern material is actually almost ring porous. So you actually have these this concentration of vessels at the growth rings. The rays are similar widths, but the ray height is considerably different. Instead of being on multiple millimeter scale, you're lucky if it gets to 0.5 millimeter. So that's considerably shorter. One similarity, of course, is the fact that you have both scalariform perforation plates in both of them. There is Eucalyptoxylon from the Kurau gravels. This is that material that I showed you right up at the start when I was talking about how you actually use the inside wood database to actually determine what a wood might be. It has solitary and very large rays. These vessels. These vessels can be 200 microns in diameter, which is very large for New Zealand. Uniseriate rays, procumbent rays, these tiny wee little intervessel pits, as well as vessel ray pits and tracheid pits. We know that we have tracheids around the vessels. We have Casuarinoxylon. This Casuarinoxylon is pretty typical for a casuarina. It has tall rays which are multiseriate. They can sometimes be aggregated. You can see how thick the rays are in this transverse section here. You have mostly solitary vessels, and you have a kind of almost random selection of ray features, ray cells. Myoporum from the Ashley River. This one is one of the ones whose identification has been queried, but that's all right. It has vessels which are mostly in less than 4 radial alignment, with this really nice kind of halo of axial parenchyma around them. It has, again, these tiny little intervessel pits that go between the 2 adjacent intervessels, and they have mostly 2, sometimes 3 seriate rays and simple perforation plates. This Melicytis material from Karatani is actually really interesting to look at. It's got a really on the hand specimen, it's got a really large kind of outer growth of calcite on it, and it was found in an actual lava flow. So it's a very weird sample, just in terms of its preservation, how it was preserved and where it was actually preserved. And that's before you start looking at the microstructure. The microstructure in this, these are technically aggregate rays, but I've never seen the tracheids and fibers in this have such an oblique endpoint coming down and cutting through this ray. There's really genuinely nothing like it in the modern, as far as I have seen, in the modern wood flora anywhere in the world, having this kind of massively oblique, almost horizontal tracheid or vessel. Unfortunately, though, the transverse face is a little bit squished, and so you can't really easily see the nice shape of the vessels. And so in the transverse face, they all look like they're kind of solitary, but when you look at them in the other directions, the radial and the tangential, you can actually tell that they're almost always in radial groups. You can actually tell that they are quite closely following alongside each other and bordering each other. They have both scalariform and simple perforation plates, which is also an uncommon feature. Right, moving on, and we're just going to talk about the Gymnosperms. So, Tom, I realise that we're getting towards the end of the talk. Araucarioxylon has a continuous record. The Eucalyptoxylons have a continuous record in New Zealand since the Jurassic. From originally only a Cretaceous start, the Planoxylon, we now know has been in New Zealand since the mid-Jurassic, which is a considerable increase to the time that this specific genus has been in New Zealand for. We have the first Cretaceous record of Casuarinoxylon from the mainland of New Zealand, as well as the first record of Taxodioloxylon from the mainland of New Zealand. And Podocarpaceae, we have the first record of Protophylloxylon and other records of Podocarpaceae from Cenozoic strata. We have, for the first time, a fossil legume wood from New Zealand. Now, during the mid-Eocene, which is when the sample was found, the legume family was undergoing a huge degree of variation and diversification and evolution. So the fact that we have a legume which is completely different to any of the ones that are alive today, like Sephora, like Kowai, is possibly not surprising, but it's nice to know that we had them. The Araliaceae from the subantarctic Auckland Islands. Now, this material, Aralia lancewood, is actually alive on the Auckland Islands today. It's one of the only 3 tree-forming species on the islands today. So it's nice to know that we've probably had it there since at least the kind of late Miocene. This is the first record of Myoporum wood from anywhere in New Zealand. It's one of the only actual fossils of the genus itself. And this is only the second record of Malacitus from Miocene strata. And that's some of the oldest Malacitus that is from New Zealand as well. Eucalypts, as I said earlier, may have actually had quite an extensive range in southern Zealandia, from kind of that North Canterbury area to central Otago. And this material is really common, and some of the samples are actually really large. So we might have had quite a large standing forest filled with eucalypts at one point. Previously, eucalypts have only really been known from really poorly preserved leaf impressions as well as disputed fruit material. Casuarinoxylon was identified from the Burnside quarry here in Dunedin. It's previously been identified from material in Southland. Casuarina and the other Casuarinaceae genera are actually quite well known from New Zealand. We have a huge amount of pollen in the Miocene deposits. It's just one example, as well as seed cones and the like. These two families, are both extinct in New Zealand, but are present in Australia and other neighbouring countries. So on to the maths part. Now, I do apologise. We had a nice 40 minutes worth of really nice photos, and now we're going to go on to something that only looks like maths. So paleoclimate reconstruction, I looked at fossil wood data to estimate paleoclimate parameters. I used 2 methods, the coexistence and Angiosperms physiognomy. The coexistent approach has been applied to New Zealand material previously because it doesn't have to use just wood material, whereas the Angiosperms physiognomy specifically uses wood, and so it had not really been done on New Zealand material in the past. For the coexistence approach, I used 2 different sources of data, the Paleoclimate database and the Kew Plants and Wood online database, and they resulted in, from the Landslip Hill genera, a paleoclimate estimation of somewhere between 18 and 25 degrees, both of them. Previous estimates from the Landslip Hill material had given estimates of between 17 and 21 degrees, so it kind of falls within the range. Excuse me. I focused more on Angiosperms physiognomy because it hasn't really been done in New Zealand before. So physiognomy means the general form or appearance of something. And I can use the incidence, so essentially the percentage of species in an area that have a feature So the incidence of a form, shape, or size of Angiosperms wood anatomy to estimate a climate parameter such as mean annual temperature, mean annual precipitation, and others. Two sets of equations have been derived by previous authors to do this from places elsewhere in the world, the Northern Hemisphere, in America, and also Europe. These are Wyman and the Martinet Cabrera and Cephalosporas. Probably messed those names up a lot. I apologize. And in total, there are 31 equations, and ideally, I would use 25 morphotypes from one site. Now, this did not happen, unfortunately. So, stepping right back, we decided that I should put together a modern feature database of all of the New Zealand woody species. And this was basically so that I could actually see if these 31 equations would actually work in estimating paleoclimate parameters to any sort of relevance. And so I put this database together, tested the equations on the entire species list, as well as specific species lists for 6 sites, 3 in the North Island, 3 in the South Island. And I basically found that I got a mean annual temperature estimate anywhere between something like 2 degrees and 40 degrees for the New Zealand average. So 40 degrees and 2 degrees. Now, wouldn't it be really nice if the modern New Zealand had a mean annual temperature of 40 degrees? We'd all be on fire. So few of those estimations were any sort of reliable, and the equations for anything other than that were even less so. We have a mean annual precipitation estimate of 5,000 millimeters for the entirety of New Zealand. Now, that's about how much rain the West Coast gets in a year in some places. 5 equations, number 11, number 22, 23, 24, and 25, were actually able to somewhat estimate paleoclimate to some sort of accuracy. And so those ones I then took and tested on fossil data, the results of which you can see here. Now, remember we were talking about the New Zealand Miocene having a mean annual temperature of somewhere between 17 and maybe 21 degrees. You can see that these ones are quite heavily underestimating that. Now, there's a couple of different possibilities as to why this is. Firstly, I said that I needed to work on at least 25 morphotypes from one site. Well, we've only got about 17 or so Angiosperms morphotypes from the entirety of New Zealand. That's considerably less, even just from all of New Zealand, than what we actually need. Another thing that I wanted to look at was the incidence of these features in the New Zealand angiosperm flora, because when I was looking at them, I was thinking some of these cannot be right. We have some features which are almost entirely feature in almost every single species, and we have some features which are entirely absent from the New Zealand flora. So have we got a relict flora from the Miocene in the modern times, or have we had extinction of species with certain features? So to test this, I did a PCA ordination on this material. I essentially went and I got the incidence of all of the features from the rest of the world, and I put them into a big spreadsheet based on the Brasier-Franklin regions, which are the regions that the Insight Wood database uses. And the result of that was actually quite interesting. New Zealand was shown to be completely separate from every other place in the world. We had a really nice kind of grouping you can see here of most of the warmer regions, another grouping of most of the colder regions, including North America, temperate Asia, and the Mediterranean. Now, sure, Mediterranean is supposed to be warm, but to be fair, South America, which is cold, is in the warm one, so I don't know what's going on there. And then New Zealand is sitting off all by itself. And the features that cause that difference are the presence of these large 10 seriate rays, which are in 22% of New Zealand's angiosperms. Now, the next highest one anywhere in the world is about 17%. So the presence of these really large rays in New Zealand is incredibly common. And the next most important is these homocellular rays. So these are rays that have all the same kind of cell type. So they're all either upright or they're all what's the word I'm looking for? Lying on their side. And the New Zealand species, we had 13.1%. Now, this is a lot lower than anywhere else in the world. Most of them are quite a bit higher. Heterocellular rays, so rays of different cell types, and septate fibers are also quite important in determining this. Then I thought, well, okay, I can probably use the Miocene fossil data set to see if the New Zealand strata, the New Zealand wood anatomy is still similar to its Miocene counterpart in New Zealand, to answer that question about whether we have a relict flora. And it kind of almost looks like we do. The Miocene fossil from New Zealand data actually plots really closely to the New Zealand, modern New Zealand. However, we do only have a few morphotypes. We've only got that less than 20 morphotypes. So we need to do more just basic finding Miocene angiosperms, sectioning them, and actually looking at them. So, summary, 24 sites throughout southern Zealandia were looked at, from the South Island to the Chatham Islands to the Auckland Islands. Somewhere between 75 and 100 samples were sectioned, but not all of them were identified because they weren't particularly well preserved. Gymnosperms are really common. The Araucarioxylon have a continuous record in New Zealand since the Jurassic at least, possibly a little bit longer. Planoxylon, Casuarinoxylon, Taxodioxylon, Protophylloclinoxylon are all present in Mesozoic strata. And while the angiosperm is rarer, it is also more diverse and includes both locally extinct material and locally extant material. The paleoclimate material wasn't really a success, but we got that really interesting output that the material, the New Zealand woody flora, doesn't really reflect any of its neighbours or any other place in the rest of the world. So there's lots of room for future work. We can always section more material. Ideally, at some point, I would find an angiosperm from the Cretaceous, because that's when they were kind of starting to first develop and evolve. And ideally, we'd do more in-depth studies at sites with this with a lot of well-preserved material like Pitt Island in the future. That is my talk. Thank you very much for coming along, and I will let you ask me some questions.